In last lecture, we have learned about prokaryotic cell, and today we are going to start a eukaryotic cell, in which we will learn today a cell membrane of eukaryotic. The eukaryotes include all the protists, which are unicellular organisms, plants, animals, and fungi. That means all these four groups are included in the eukaryotic groups. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm. Means cytoplasm will be compartmentalized, through the presence of a membrane bound organelles. Inside the cytoplasm, there is the presence of a membrane bound organelles. Eukaryotic cell possesses an organized nucleus with a nuclear envelope, and that's why only we are consider it as a eukaryotic cell. As you know, what is the difference between eukaryotic cell and a prokaryotic cell? Basic difference between eukaryotic cell and a prokaryotic cell is the presence and absence of a nuclear envelope. If nuclear envelope is present, then it is considered as a eukaryotic cell, and if not present, then it is considered as a prokaryotic cell. So now we are talking about eukaryotic cells. So here nucleus will have a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotors, and skeletal structures. Their genetic material is well organized, and that's why you will see here or observe here a chromosomes. Plant and animal cells are different as the former possess cell wall. Means plant will different from the animal. How it is different? Because plant cell will have a cell walls, plastids, and large central vacuole, as we have already discussed in previous lecture. What is the difference between animal cells and plant cells, which are absent in animal cells? So you know very well now what is the absent in which structures are absent in an animal cell? Cell walls, plastids, and the large central vacuoles. Whereas these all structures are only found in the plant cell. On other hand, animal cell will have a centriole, and the centriole will be absent, totally absent in case of a plant cell. So animal cell have a centriole, which are the absent in almost all plant cells. So this is the difference between animal cells and the plant cells. Today we are going to discuss about cell membrane of a eukaryotic cells. The detailed structure of the membrane, that is a cell membrane, was studied only after the advent of the electron microscope in the 1950s. Here in this diagram you can see an electron microscope. All the living cells covered by the thin, delicate, elastic, selectively permeable, we will see what is the selectively permeability, and living boundary, which is called cell membrane. The name cell membrane was given by Nageli and Kramer. And same structure was called as a plasma lemma by another scientist, that is the J.Q. Plow. So two name will be was given at that time. Cell membrane and plasma lemma. So cell membrane name was given by Nageli and Kramer. And plasma lemma name is given by J.Q. Plow. Or a biomembrane or a plasma membrane. So many other names are also there. So we can say cell membrane also, plasma lemma, biomembrane, or a plasma membrane. These all are the synonyms of the cell membrane. Especially in human red blood cell, that is RBCs, enabled the scientists to study possible structure of a plasma membrane. First cell which is studied for the cell membrane was a RBC. These studies showed that the cell membrane is composed of lipids that are arranged in a bilayer. In the cell membrane, lipids are arranged within the membrane with the polar head towards the outer side. Here in this diagram, you can see this is a red color head-like structure, pointed structure, is a hydrophilic polar end which is situated outside. And the hydrophobic tail towards the inner part. Here you can see this is yellowish color tail-like structure is a hydrophobic non-polar end, and which will be inner side. So outer side hydrophobic head, and inner side that will be hydrophobic tail. And that will prepare a phospholipid bilayer. These ensure that the non-polar tail, that is a hydrophobic tail of a saturated hydrocarbon, is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglycerides. Later, Biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate along with the lipids. Okay, or we can say phospholipids. So along with the phospholipid inside the cell membrane, there will be a presence of proteins and carbohydrate both. Here you can see in the diagram, this is the protein which is embedded inside the lipid layer. Okay, so it is found that after the biochemical investigation, it is found that, and it is confirmed that along with the phospholipids, proteins and carbohydrates are also a component of any cell membrane. The ratio of a protein and lipid varies considerably from cell to cell. Means from cell to cell it is be different. In human being, the membrane of a erythrocyte, that is RBC, has approximately 52% of a protein. Okay, especially when you talk about erythrocyte, that is RBC, it will have approximately 52% of a protein and 40% of a lipid. Depending on the age of extraction, membrane proteins can be classified as an integral or a peripheral. Here you can see clearly in the diagram, this is the lipid membrane. Okay, and here you can see this molecule of a protein is embedded throughout the lipid membrane. Okay, and that's why if the protein is throughout, it is embedded, then it is called integral membrane protein. Okay, and if this protein is not throughout the integrated, then it is called peripheral protein. These both are a peripheral protein. So by this way you can divide proteins into two groups, integral and peripheral. Peripheral proteins lie on the surface of the membrane, you can see here. Okay, while the integral proteins are partially or totally buried in the membrane. You can see the clear vision in the diagram. After the discovery of a cell membrane, number of scientists had given some models to explain a structure of a cell membrane. Okay, so now we will discuss which type of model is given by which scientist and whether it is accepted or rejected. So first model, let me discuss about sandwich model, or it is also called trilamellar model, and it was given by Dawson and Danieli in 1935. So according to this model, what they have explained? According to this model, the plasma membrane is made up of three layers. Okay, in which bimolecular layer of a lipid, okay, is sandwiched between two single layer of a protein. Means two layers of a protein was there, and in between a lipid layer was there, and that's why it is written a lipid layer is a sandwich in between the protein layers, and that's why it is considered as a sandwich model. According to this model, each protein layer is a 20 angstrom thick and bilayer of phospholipid is a 35 angstrom thick, and thus the total thickness of the layer is a 75 angstrom. And the phospholipid molecule called amphipathic molecule. What is the amphipathic molecule? That means it have a hydrophilic and hydrophobic as well as the structure. Means due to presence of two type of parts, it having a hydrophilic head and hydrophobic tail, and that's why such a layer is considered as an amphipathic molecule. The hydrophilic head of the phospholipid bind with the protein layer by hydrogen and ionic bonds. According to the model, hydrophobic tail of a phospholipid are attached to each other by van der Waals bond. Okay, or van der Waals forces, and this is according to sandwich or a trilaminar model, which was given by Dawson and Danieli in 1930. Now let me summarize by using this diagram. What is a sandwich model? A sandwich model which is given by Dawson and Danieli. Okay, according to that, what is there? This is the protein layer. Okay, here also one protein layer that will be create a hydrophilic zone. Okay, and in between there will be hydrophobic zone, and this is the lipid layer which is sandwiched in between protein layers. Okay, so it is the sandwich model. That's why it is called sandwich model of Dawson and Daniel. Next model is a unit membrane model, and it was given by Robertson in 1959. According to this model, all the cellular and organellar membranes are structurally and functionally similar. Okay, and only difference in between that are only chemicals and size. Both above model, whether we are talking about sandwich model or unit membrane model, both were rejected. because they fail to explain the elasticity related principles and selective permeability related plasmarema. Okay, that means they cannot explain elasticity as well as selective permeability membrane related function of a plasma lemma. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Okay, and meanwhile chemical studies on the cell membrane, especially in human red blood cells, okay, enable the scientists to deduce or study the possible structure of a plasma membrane. After the rejection of a sandwich model and unit membrane model, one new approved model was represented or proposed by Singer and Nicholson in 1972, and that was widely accepted as a fluid mosaic model. So let me try to understand what is a fluid mosaic model. This is the latest and most widely accepted model for the structure of a plasma lemma or a cell membrane. So first of all, main component of any cell membrane is a phospholipid. So phospholipid is the main component of the cell membrane because it forms a continuous structural frame of a cell membrane. So it is considered as a main component. The studies showed that the cell membrane is composed of lipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. Okay, and these ensure that the non-polar tail of a saturated hydrocarbons is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglyceride according to this model. Second important component of any cell membrane is a protein, and there are of two types. That is, first type is the integral protein and second is the peripheral protein. So integral protein is also considered as an intrinsic protein and peripheral protein is also considered as an extrinsic protein. So let me try to understand what is the integral proteins. These proteins are tightly bound with the phospholipids. Okay, and thus they cannot easily remove from the membrane because it is tightly bound. Integral proteins are of two types. Further, it will be divided into two types, partially buried or totally buried. Okay, some integral proteins which are totally buried through the complete thickness of the membrane. And second is the peripheral protein. And these are the superficially arranged on the outer side and it can be easily removed or separate by the easily methods. Okay, these protein have enzymatic activity. Means some of these protein may act as enzymes also. Plasma membrane have approximate 30 type of enzymes. Means it will the proteins which are present in the plasma membrane will act as a enzyme, and they are found that 30 type of enzymes are acting as enzymes. Means 30 type of proteins are acting as enzymes, in which ATPase is major. Okay, ATPase, that is a ATP hydrolyzing enzyme, is a more important. ATPase enzyme help in the active transport across the cell membrane. The fluid nature of the membrane is also important from the point of view of functions. Means all the functions of a cell membrane is just due to their fluid nature, and that function includes cell growth, formation of intercellular junctions, secretions, endocytosis, cell division. These all functions is due to fluid nature of a cell membrane. One of the most important function of the plasma membrane is the transport of the molecule across the cell membrane. That is called transmembrane transport. Okay, this is very important phenomena because anytime whenever you observe the cell, something will be come out of the cell and something will be added to the cell. This is the very important phenomena of any cell. So transport of a molecule across it is possible just due to cell membrane. The membrane is a selectively permeable to some molecules. Means it will allow some molecule to come inside and outside. Okay, present on either side of it. Many molecule can move briefly across the membrane without any requirement of energy. So if movement occur without the requirement of energy, then this type of movement is considered as a passive transport. So by this way we can divide transport in two types. Okay, transport can be divided into two groups. One, active transport, and second is the passive transport. When it is considered as active transport, when energy is utilized at that time, it is considered as active transport. And when energy do not utilize at that time, it is considered as a passive transport. Neutral solute may move across the membrane by process of a simple diffusion. Okay, and along the concentration gradient. That means from higher concentration to lower. So here you can see some molecules from higher concentration to lower concentration, it is moving. Okay, without using any energy. So this is one kind of a passive transport. Here also you can see from higher concentration some molecules are moved by using a protein molecule. So here without using protein molecule and energy, this type of movement is considered as a simple diffusion. And if without using energy they are just using the protein molecules, okay, such a important type of transport is considered as a facilitated diffusion. So passive transport can be divided into two groups: diffusion and facilitated diffusion. Let me repeat, what is a passive transport? Passive transport is the transport across the cell membrane without using any energy, and that can be divided into two groups: simple diffusion and facilitated diffusion. So what is the difference between diffusion and facilitated diffusion? In simple diffusion, only from higher concentration to lower concentration molecule will be transported, whereas in the case of facilitated diffusion, from the higher concentration to lower concentration with the help of protein, molecule will be transported. Another important transport is of water, and that is called osmosis. So let me try to understand what is osmosis. Water may also move across this membrane from higher to lower concentration, and movement of a water by diffusion is called osmosis. Okay, so you know very well any solution is made up of solute molecule and solvent molecule. And if you are talking about solute molecule to transport across the membrane, it is called diffusion. Okay, from higher concentration to lower concentration. And if you are talking about solvent molecule to move across the cell membrane from higher concentration to lower concentration, it is considered as a osmosis. Now let me try to understand the concept diffusion and osmosis by using this diagram. So diffusion and osmosis, how we can define diffusion and osmosis, and how we can differentiate between diffusion and osmosis. All of you know any solution is made up of solute and solvent, right? So if you are explaining a movement of a solute from higher concentration to lower concentration, so such a movement is considered as a diffusion. Means if you are applying the movement of a solute from higher concentration to lower concentration, it is considered as a diffusion. And if you are explaining the movement of a solvent molecule from higher concentration to lower concentration, that is considered as a osmosis. So this is the definition of a diffusion, that movement of a solute molecule from an area of a higher concentration to an area to a lower concentration, that is considered as a simple diffusion. And if you want to define osmosis, then you just replace this word solute to the solvent. Okay, means let me repeat, what is osmosis? Osmosis is the movement of a solvent. You just have to replace this word with the solvent. Okay, means osmosis is the movement of a solvent molecule from an area of a higher concentration to a lower concentration. So if you want to explain a movement of a water at that time, you have to use the terminology osmosis. Few ions or molecules are transported across the membrane against their concentration gradient. That means from lower concentration to higher concentration. It is very common to transport from higher concentration to lower concentration, but here what happened? It is against the concentration gradient. That means from lower concentration to higher concentration. So here you can see a low concentration molecule will transported towards the higher concentration, and that's why it is against the gradient, and that's why here ATP as the energy will be utilized. So this is the definition, that if transportation occur with the help of energy utilization, that is considered as a active transport. So these here you can see, this is the molecule, you can see here lower concentration, from lower concentration it is transported towards the higher concentration with the help of ATP. So this is called called active transport. Such a transport, it is an energy dependent process because without energy this is not possible. Okay, in which ATP is utilized and it is called active transport. Such an example of active transport is a sodium potassium pump. So now let me summarize by using this diagram. Here you can see this molecule is transported across the membrane without using any energy and protein. So this is called simple diffusion. Okay, and this molecule is transported across the membrane without using energy but with the help of a protein, then it is called passive transport. And this passive transport can be divided into two groups, a channel mediated diffusion and carrier mediated diffusion. These both are a passive transport because here also ATP is not utilized or energy is not utilized. Okay, and if ATP is utilized during the transportation from low concentration to high concentration and ATP utilization occur, such a transportation is considered as an active transport. Facilitated transport or a protein mediated transport can be divided further into three groups. Here you can see that is a uniport, symport and antiport. Okay, so let me differentiate in between these three. So uniport is the transport of a facilitated transport in which one molecule will be transported across the membrane by using protein. Okay, so if only one molecule is transported across the membrane, that is considered as a uniport, and if two molecules together parallelly transported across the membrane in the same direction, then it is considered as a symport. And if two molecules transported across the cell membrane but not in the same direction, it is in the opposite direction, then it is considered as the antiport. Means here you can see one molecule A will be transported from outside to inner side and another molecule simultaneously, or we can say parallelly, transported across the membrane from inner side to outside, but direction is different. Okay, if direction is different, then it is considered as antiport. If direction is same, it is considered symport. And if only one individual molecule is transported, it is considered as a uniport. This is the difference between uniport, symport and antiport. Now let me summarize all type by using this tree. Here you can see transmembrane transport can be divided into two groups, passive transport and active transport. Okay, so what are the passive transport? Passive transport are the transport in which energy utilization not occur. So it is considered as a passive transport and it is always from higher concentration to lower concentration. This is the definition of passive transport. And what is the active transport? Active transport is the transport in which energy is utilized, okay, and it will be always from lower concentration to higher concentration. This is main difference between active transport and passive transport. Passive transport can be further divided into two groups, simple diffusion and facilitated diffusion. If without using protein diffusion is occur, then it is considered simple diffusion, and if protein is utilized during the transportation, then it is considered as a facilitated diffusion. And that can be further divided into two groups, channel proteins and passive carrier proteins. Okay, and further facilitated diffusion can be divided into uniport, symport and antiport also. And active transport, during active transport there are no types of this. Okay, only definition is that during the transportation energy will be utilized from the low concentration to high concentration. This transport will be so this is your homework for today's lecture, and just write down your homework in your notebook.